Hello, grade 10. Today we are still in section 6, chapter 8, gene expression and regulation. First, we will review what we took yesterday. We were talking about the gene expression in prokaryotic cells. We said that prokaryotic cells turn genes on and off by controlling transcription. Okay? Now we said that the gene in the prokaryotic cells have an organized in a system which called operon, and this operon contains promoter, operator, and the genes that will be coded for the synthesis of a certain protein. Now, promoter and operator are segments at the DNA. The promoter allows a gene to be transcribed, and this helps the RNA polymerase to transcribe the mRNA. Now, the operator is part of DNA that turns a gene on and off. Okay? So, the most important thing you have to know here that the operon is a system or the gene, organization of the gene in the prokaryotic cells, which includes a promoter, operator, and one or more structural genes. Yani sometimes we have more than one gene that controlled by one promoter and one operator. Okay? Whereas in the eukaryotic cells that is not happening. Each gene has specific promoter. Alright, so operons are most common in prokaryotes, and you will take example of operon, which is the lac operon, which is specific for the lactose. Okay, this example will explain for you the gene regulation in the bacteria. Okay, now the lac operon has 3 genes that codes for the synthesis of enzyme that break down the lactase. Okay, so lac operon, as we said here, has 3 genes. These genes code for the enzyme, which is lactose, to break down lactase, which will break down the lactose. Now, this operon can switch on and off depending on the absence or presence of the lactose in the bacteria's body. Now, if there is no lactose, the lactose is not present, it's absent. No need for this enzyme to be produced or synthesized, and we said the enzyme is the protein. So what will happen here? We will see the promoter and the operator. Promoter, this sequence of DNA will operator. The operator part will be bound to a protein which will block the RNA polymerase from transcribing the DNA. So that means the lactase will not be synthesized. The gene will not code for the enzyme or the protein to be synthesized. Otherwise, if lactose present, this will cause this lactose to bind to the repressor. Repressor is the operator with the protein, okay, and it will change its shape, so it will leave the strand of DNA, and this will allow the RNA polymerase to start transcribing and forming mRNA. The mRNA is growing here. Okay, so in prokaryotic cells, gene expression regulation can happen by switching on or switching off an operon. Okay, so now in the eukaryotic cells, as we know that every body cell in an organism has same set of DNA, same number of chromosomes, but your cells are not all the same because you have different types of cells. Each cell has different function. So what makes the cells differ? Cells differ from each other because different sets of genes are expressed in different types of cells. Not all the genes of the DNA is expressed in all the cells. Only the genes that is needed by the cell is expressed. So Now, eukaryotic cells can control the process of gene expression at many different points because of their internal compartments and chromosomal organization. As you see here, we have a chromosome, and this chromosome is made, if you look at the chromatin fiber, it's made of DNA that wrapped around the histones. Some parts of this DNA are very condensed and some are loose. So not all the genes could be expressed in all the DNA strands. Now, eukaryotic cells regulate the gene expression at many points, okay? And how this happen? It's controlled by first three things. The first thing is regulatory DNA sequences. Regulatory DNA sequences are segments on the DNA that regulate the gene expression. And these could be enhancers, promoters, or silencers. These are the regulatory DNA sequences are nucleotide sequences that make segments on DNA. And also the other thing that regulate the gene expression are the transcription factors. Transcription factors are proteins. Each type of cell have different types of transcription factors. Like مثلا عندي أنا gland cells, okay, that release the saliva, which have amylase enzyme. They have transcription factors for the amylase. While the liver don't have these transcription factor specific for amylase production. So each cell has different transcription factors from others. And these are mainly proteins. Okay? And of course the other part, which is the RNA polymerase. We will stop till here and tomorrow we'll continue. Thank you and take care.